Welcome to This Explainer. I'm Dr. Chem, and today we're putting on our lab coats to explore the fascinating science of lipid detection. We're going to see exactly how scientists isolate, extract, and analyze the hidden fats inside our everyday foods, from a simple glass of milk to a handful of nuts.
Here is our roadmap for today. First, what exactly are food lipids? Then, we'll look at the sample preparation challenge, the science of extraction, qualitative lipid testing, and finally, advanced lipid analysis. Let's get right into it.
Starting with Section 1, what are food lipids? It's time to unmask the molecular structure of fats.
To understand how we actually extract fats from foods like milk, cheese, or meat, we first need to know our target. What exactly are we hunting for here? Well, at their core, lipids are a really diverse group of molecules. This includes fatty acids, phospholipids, and steroids like cholesterol. But here is the absolute crucial point. Lipids have a very specific, kind of stubborn relationship with water. They are largely non-polar. That means they are completely insoluble in water, but they'll happily dissolve in non-aqueous organic solvents like chloroform. This fundamental chemical quirk, that's the basis for almost everything we do in lipid detection.
Lipids have this really interesting amphiphilic nature. It's almost like they have a split personality. These molecules often feature a water-fearing or hydrophobic zone alongside a water-loving hydrophilic outer layer. This dual nature dictates exactly how they hide inside cells, and consequently how we have to coax them out.
Moving on to Section 2, the sample preparation challenge, or why we can't just squeeze the fat out.
Because lipids are so stubborn and trapped inside complex food matrices, we have to meticulously prep our samples. Take hazelnuts, for example. We can't just press them and hope for the best. By mechanically reducing the particle size, literally just chopping them up, we drastically increase the surface area. This ensures that when we add our chemical solvents later, they can penetrate those food tissues highly efficiently and grab those hidden fats.
But physical chopping isn't always enough. In foods like dairy, bread, and animal products, lipids are actually chemically bound to proteins and carbohydrates. They're locked in tight, so a process called acid hydrolysis is required here. By refluxing the sample with hydrochloric acid, we can literally break those strong covalent and ionic bonds, freeing the lipids into an easily extractable form.
So let's see how this all builds into our standard 3-step sample preparation protocol. First up, we remove the water through pre-drying, because organic solvents really don't mix well with moisture. Second, we reduce the particle size. And third, if necessary, we hit it with that acid hydrolysis. Boom! Now our food is perfectly primed for extraction.
Which brings us to Section 3, the science of extraction, flushing out the hidden suspects.
With our sample perfectly prepped, one classical and highly effective method uses an automatic sock-slit extraction machine. It's an incredible piece of lab equipment. It systematically immerses the sample in an organic solvent, usually running at around 180 degrees Celsius. After a thorough washing phase, the organic solvent is distilled and separated, recovering the pure oil. It's literally chemistry in motion.
Now, the core rule of solvent extraction driving these machines is actually pretty simple. Polar liquids, like water, actively repel non-polar solvents, like hexane. It's the old oil and water don't mix rule. By using a non-polar solvent, we ensure it only interacts with and dissolves the non-polar fats, completely ignoring the water-loving proteins and carbohydrates left behind in the food.
Another fantastic technique is the Bly and Dyer method. We use this heavily for samples with high water content. Think fish muscle, for instance. It uses a rapid ternary solvent system, which is just a fancy way of saying a 3-part mix of chloroform, methanol, and water. When mixed, it intentionally breaks into 2 distinct phases. As you can probably picture, the non-lipid molecules stay trapped in the upper aqueous phase, while our target lipids drop perfectly into the lower chloroform phase, totally ready to be collected and evaporated. All right, Section 4, qualitative lipid testing, finding our first clues in the forensic lab.
So we've extracted what we think are invisible fats into our solvent. But how do we actually prove they're there? Simple preliminary tests can give us immediate, satisfying answers. Take the emulsion test. If you mix an oil drop with a little water, a distinct, cloudy white layer of suspended fat globules forms at the top of the solution. If the solution stays completely colorless, well, you don't have lipids. It's a really simple, visual confirmation right there in the test tube.
Beyond emulsions, we rely on an entire toolkit of qualitative reactions. There's the classic spot test, also known as the grease spot test, where a sample leaves a translucent, greasy spot that completely penetrates filter paper. There are solubility tests, And then there are dramatic chemical reactions, like Salkowski's test, which uses concentrated sulfuric acid to turn cholesterol into this incredibly vivid red bicholesteridine sulfonate. It's like magic, but it's science.
We can also perform highly specific qualitative color tests. If we're on the hunt for cholesterol, the Libermann-Burchard reaction is an absolute favorite. By adding acetic anhydride and a few drops of sulfuric acid to a chloroform extract, a successful test yields a striking rose to bluish-green solution. These dramatic color changes, that's the biochemical proof that our extraction actually worked.
So these colorful qualitative tests confirm the fat is definitely there. But what if we need to know exactly what kind of fat we're dealing with? Are we looking at trans fats? Hydrogenated fats? Answering that requires us to pivot from these qualitative color changes to some seriously advanced, highly precise methodologies.
Which leads us to Section 5, Advanced Lipid Analysis, Precision at the Molecular Level.
To measure those specific health-impacting components hiding inside our lipid extract, we turn to the gold standard of modern food safety analysis, gas chromatography. But here's the catch. We can't just pour our fat directly into a high-tech machine. It requires a rigorous three-step chemical preparation. First, the extraction we just talked about. Second, saponification, which breaks the fats down into free fatty acids. And third, derivatization. This step chemically converts the fatty acids into highly volatile fatty acid methyl esters, or FAMEs, making them thermally stable enough to actually survive the gas chromatograph.
Once they're derivatized into FAMEs, the gas chromatography machine works its magic. It meticulously separates the components based on really slight differences in their physical and chemical properties. This machine is so precise, it can identify up to 37 common fatty acids, generating incredibly detailed data on the exact molecular composition of the fat content in a piece of pork, a glass of milk, or a handful of grain.
The science of lipid analysis reveals an entirely hidden world right inside our pantries. From using acid to break molecular bonds, to relying on polar repulsion to isolate oils, all the way to vaporizing methyl esters in a gas chromatograph, it proves that our everyday meals are true biochemical marvels just waiting to be explored. So next time you consume a piece of cheese or fry a potato, ask yourself, what complex, microscopic chemistry is hiding inside that simple bite? Thank you for joining me on this explainer, and keep questioning the world around you.